Good morning, everyone. This is Kameshwari from Nallana Simhareddy Education Society Group of Institutions. Today, we are going to discuss about Environmental Engineering Laboratory. In that, the experiment is dissolved oxygen. What is dissolved oxygen? Any idea? What are the operators required for determination of dissolved oxygen? So to find out the amount of dissolved oxygen in the given sample of water, to find out the amount of dissolved oxygen in the given water sample. And the operator required for determination of this test is two types. One is DO meter and we require beakers. Otherwise we can go for photospectrometer and samplers. So what's the procedure? First, before going to procedure, what is the need of dissolved oxygen? So here, dissolved oxygen is essential to the survival of aquatic organisms. Naturally present dissolved oxygen enters the water. When plants photosynthesize, wind and wave action also cause oxygen from the air to dissolve into water. To survival of any animals, plants or human beings, we require oxygen. Dissolved oxygen is consumed by aquatic animals and by the oxidation or chemical breakdown of dead and decaying plants and animals. The concentration of dissolved oxygen in natural water can range from 0 to 14 ppm. Natural water ranges is 0 to 14 ppm and affected by temperature and salinity. Here we required water samples. We can take either drinking water and surface water, domestic water, industrial water, etc. The range is generally between 0 to 12 ppm dissolved oxygen. So here this method use the agile modification of the Winkler method with a colorimeter determination of the yellow iodide produced from the reaction with the dissolved oxygen. So here generally the normal water is 5 to 9.5 mg per liter and warm water 5 to 6 and cold water 6.5 to 9.5. So this is the general description of dissolved oxygen. So now in this test, in this session, we are discussed regarding photospectrometer. So what's the procedure? The first step is press on button to turn photospectrometer on and scroll to and select program test. And in this photospectrometer, it consists of a more number of tests. So, and then it will give an alphabetic order. So, we can select a dissolved oxygen D, order D. So, scroll to and select all test or another sequence containing 39 DO from testing menu. We can select, scroll and select all test and we can go for 39 DO. Scroll to and select 39 DO from menu. Now rinse the clean tube with untreated soil, sampler water and fill to the 10 ml line with the sample. The sampler is taken, and then we can fill the water may be tested in the sample. That sample will be poured in a 10 ml line mark. This tube is the blank. Insert tube into the chamber, close lid and select scan blank and fill second tube to the 10 line with a treated fixed sample. This tube is the sample. So remove blank from the spectrophotometer, insert sample tube into chamber, close lid and select scan sample, record result. So here press and hold half button to turn photospectrometer half, or press exit button to exit to a previous menu or make another menu selection. Now we can turn to laboratory, how to determine dissolved oxygen in our environmental engineering lab. Hi students, in this session we are in environmental engineering laboratory. Today we are going to see how to calculate dissolved oxygen present in the given water sample by using the photospectrometer. Now how to operate photospectrometer? First here program test, click on program test. Now sequence 1, 2, 3 and after that all test, click on all test by using downward arrow, enter. Now here today we are going to discuss dissolved oxygen. What's the number dissolved oxygen? Here alphabetic order will be there. By applying, by click on downward arrow, go for D. The serial number 39. Slowly press the downward arrow. So finally we will get 39 serial number and DO. Now open the lid. First we can place the blank sample. What's the blank sample means? What's the blank sample? Blank sample means distilled water. So first clean the sampler carefully and click on the enter. First place the sample, blank sample into the photospectrometer. Now click on, close the lid, click on the enter. Now scan sample, whatever may be sample required for, we can know the dissolved oxygen, that particular sample taken out from the blank sample, and we can insert another sample. We can know that particular sample dissolved oxygen. Insert carefully and close the lid and click on enter. So here the value will be 0 mg per liter.